All right, so we will continue our lectures for Stat M254. As I mentioned in last lecture and in my syllabus, so for this year, I want to teach something new in the new format. So if you would like to know what I covered in previous years, you can still check the recordings from last year, and I will post them. I think I already posted them on Brulearn, so you should have access to the lectures. But for this year, what I want to do is to focus on the single cell data analysis using the R package SURAT and discuss statistical concepts within the SURAT steps. So for your final project, it will be a single cell data analysis because I'm very interested in seeing how people may do the analysis in different way to see what diversity we may have in the data analysis. So for the lectures I'm going to talk about today and on the following lectures, I will talk about statistics in SURAT steps. So that's the plan. But I will still try to cover the previous materials I covered in this new timeline. So try to reorganize things by following the data analysis. I think that may make the data analysis more in a more streamlined way, and then you can see how statistics is used in different steps of data analysis. And single cell data analysis is just one example. I think many of the ideas should be applicable to other types of genomic data analysis. That's the hope. Okay, and I will post the slides later after the class. So first of all, I want everybody to have this impression. Computation analysis is complex. So as I said, the last lecture's take-home message is to try to understand the difference between inference about the population and prediction for individual instances. That was for last lecture. And as examples for inference, you can think about hypothesis tests, and for prediction, you can think about classification and regression. So these are two supervised learning, and they focus on instances. So for today, I want you to have the message as the data analysis pipeline is complex. It cannot be automatic, saying that you just follow one tutorial, copy the code for each step, and you can get a good result. That's not the case. And anything you do during the analysis would have a role, would have an impact on your analysis result. So this is the correspondence piece I authored last year about this connection, which I found interesting about this Monty Hall problem. I think for those of you who are Americans, you might know this as a popular culture thing from a TV program and the false discovery rate in our data analysis. So this is just the opening of today's lecture. So what is the Monty Hall problem? It's a very interesting TV context. So a contestant is like an audience who participate in this game would try to guess behind 3 doors which door has a car behind and the other doors have a goat behind. The host knows the answer. Okay, so how the game works is that the contestant will randomly pick one door as the first choice. Then we know that in the remaining 2 choices, there must be a goat behind. So by the way, after the first choice, the door stayed closed. So it's not opening the door, just make a choice. And then the host will open one of the other 2 doors with a goat behind. Because the host knows the ground truth, so the door he opens will surely have a goat behind, not a car behind. Then there are 2 doors closed. The one picked by the contestant in the first round and another door. Then the contestant has a choice whether he or she wants to switch the door, swap the door to the other choice, or stay with the first choice. So that's the question. And it became a brain teaser and became a very, very big discussion in public. So the question is, will the contestant have a better chance of winning the car if he swaps in the second round, he or she swaps in the second round, or stays at the first choice? Most people's guess is it doesn't matter, including many mathematicians. The reason is people have this intuition that with 2 doors, and either one could have the car behind, then the two doors have equal probabilities of having the car, then it doesn't matter if I swap or not. However, what I'm doing here is try to draw this whole process out. And you can see that in the first step, the contestant has only 1/3 of the chance of picking the car. And if that's the case, then swapping would make him lose, will make him lose, right? And but here I'm talking about the probability of not swapping. Then staying with it, it will win. But for the other two cases, 2 out of 3 chances, the contestant may pick the goat. Then in this case, the contestant should swap. So why does it matter? The reason is when the contestant actually picks a goat in the first step, then only one of the remaining 2 doors have a goat behind. Then the host has to know the truth to open the goat door, not the car door. So basically what the host opens, is not a random pick, but based on knowledge. And that will affect the result. So even though it's counterintuitive, but the fact is it's better to swap than not to. Okay, but I'm trying to draw the second scenario to explain. It's like a brain experiment, a thought experiment, I should say, to explain why people might have the confusion. The reason is when people think about equal chance for switching and not switching, they're thinking about the second scenario where the host first pick a goat door to open. Then the contestant is left with 2 doors, one with a car, the other with a goat. Then in this case, it doesn't matter which of the other doors the contestant chooses because the 2 doors have equal chances of having a car. So therefore, the order of actions matter. Who chooses first, the contestant or the host? That will change the outcome probability. I hope this is clear to you. So if this is a new problem to you, I think you might find this Monty Hall problem interesting. It's a probability brain teaser problem, basically brain teaser problem. Basically, you need to be very careful to think about the order of actions. And this is what I take from it. And why is it related to false discovery control? So this is also what I have in the paper, in the correspondence. So a topic we will cover later today, not in detail today, it's called multiple testing correction. When you do multiple hypothesis tests and you calculate a p-value per test, the final step is to threshold your p-value for some criterion that guarantees the overall performance of your multiple testing correction. Hopefully, you're not making too many mistakes. So a popular criterion is called the false discovery proportion, defined as, is the false discovery rate. It's defined as the expectation of a proportion called false discovery proportion. So we need to first define FDP, false discovery proportion, then define its expectation as the false discovery rate, FDR. So if you briefly mention what I mentioned last lecture, frequentist versus Bayesian, excuse me. Here we are talking about the frequentist paradigm in statistics. So in the frequentist paradigm, we believe that which discoveries are true, which are false, are fixed. Okay, are some unknown but fixed ground truth. So by discovery, you can think about this as differentially expressed genes. We're trying to find differentially expressed genes between conditions. Then the true DE genes and the true non-DE genes are unknown ground truth. But every time we collect the data, we do the experiments. From our experimental data, there is this proportion. However, we don't know it because we can only say, okay, after we threshold the p-value to some threshold, say 0.001, we count the number of genes with p-values under the threshold, and that's our number of discoveries. So we would know this denominator, but we don't know the numerator because knowing the numerator requires you know the ground truth, and that's what we don't have. But the magic here is, even though FTP itself is a ratio you cannot observe, but you can somehow control the expectation of FTP, which is FDR, under a threshold. And the requirement here are twofold. First, for the p-values coming from the true non-DEGs, even though you don't know what they are, but you believe that the majority of your genes should be non-DEGs, then their p-values should be uniformly distributed between 0 and 1. And the second assumption is the independence of the p-values. So we know that the genes are correlated, not independent. So that is something we need to be very careful. It's assumption needed by the theory. But in practice, people found that when genes have some positive correlations and not too many of the genes are correlated, like the majority of genes are still kind of uncorrelated, then the FDR-based p-value thresholding may still work fine. And there are some other thresholding procedure to control the FDR by respecting the correlation, but they are in general more conservative than the ones assuming independence. By conservative, I mean that you would have fewer discoveries. You would set a smaller p-value threshold and have fewer discoveries. Okay, so the top row is the correct approach. Calculate p-values in step 1, thresholding p-values in step 2 to control the FDR. And then after you control the FDR, you have your discoveries. Then you, as a biologist, right, you want to do some experimental validation. If some of the discoveries look like interesting targets, you may do validation to pursue, to validate or to investigate their mechanism, why they are useful markers. Maybe they are some drivers. So that's the whole idea. But what could be problematic is that if you do this manual feature screening after you calculate p-values, but before you do p-value thresholding. What I mean is that if you use the p-values to sort your features, like your genes, from the smallest p-value to the largest p-value, and you arbitrarily said, okay, I will only consider the top 100 genes with the smallest p-values. Then when you apply p-value thresholding, you only consider these top 100 p-values instead of all the p-values. Then you cannot control the FDR. So basically what I'm plotting here is a simulation example in which I strictly implement the top one or the bottom one. And this is a violin plot showing the distribution of FTP, and the bar shows the average as the FDR. So I'm doing this experiment in simulation for many, many times. So each time, because it's a simulation, I know the ground truth, so I can calculate the FTP. And then I can plot the distribution of FTP and the average. You can see that for the top one, the FDR is at 0.05, which is my target, even though the FTP itself may have some variation. But for the second approach, you can see that the FTP and FDR can be way above 0.05. So this is another way I'm trying to draw the connection, because here the similarity with the Monty Hall problem is that we change the order of actions, then the result will be different. Okay, so this is the opening, and I just want to use one slide to briefly reiterate what I just said regarding multiple testing. So hopefully we'll cover this later in the class. But for now, I just want to say that the false discovery rate was a concept introduced in the statistics paper in 1995. In the frequentist paradigm, FDR is defined as expectation. Expectation is taken over the distribution of potential data sets from the same distribution, from the same population. For each data set, this ratio is called FDP. And this notation here, this angle here means the minimum between the number of discoveries and 1. So it's just a way to write the minimum. Basically, you don't want to have the dividing 0 issue. So that's why when you have a 0 discovery, you divide 1. Okay, and what I just said, the p-value based FDR control, the first and most widely used procedure is from this paper that proposed FDR by 2 authors, Benjamini and Hochberg. And their procedure is very simple as an operation. Basically, if you have M features, say M genes, you have 1 p-value per feature. you sort them from the smallest to the largest, and you have a target FDR in mind, say Q as 5%. That's the common Q value, common value for Q. And then what you did is that you compare each ordered p-value, the jth smallest p-value, to this changing threshold. So the threshold is determined by Q, say 5%, number of features, and the order. So for the smallest p-value P1, you are comparing to 1 over m times Q. Second smallest p-value P2, you compare it to 2 over m times Q. You want this inequality to hold until it breaks. So whenever it breaks, you just record the last k, largest k, such that the inequality remains to hold. And then you reject all the P1 to Pk, the smallest ones. That's how you're going to find the threshold. So basically, the threshold is determined by a consecutive order, of inequalities. So you need to have the first k inequalities to hold, k plus 1 breaks, then you reject P1 to Pk. So that's the Benjamin-Hochberg procedure to control the FDR. And the theory would require uniformity of p-values if they are coming from the null hypothesis. And later, John Story has a q-value procedure, which is also widely used in bioinformatics. It has a slight modification to the definition of p-value, of FDR, and it considers it needs a guess, a rough guess for the proportion of null hypothesis or has a for the features that are non-DEG. So you need a rough guess for the proportion. Then in the procedure, you set the proportion to your guess instead of setting the proportion to be 1, which is the case in the Benjamin-Hochberg. So Benjamin-Hochberg can control the FDR without a problem, but it's slightly more conservative than Story's Q value if you guess the proportion. For example, if the proportion is not close to 1, then their result may have a bigger difference. But if the proportion it guesses in Story's procedure is close to 1, that is, majority of genes are non-DEGs, then their results will be similar. So both procedures will require p-values. And validity here means that null p-values are uniform between 0 and 1. And high resolution means that if the p-value is small, you'd better have more digits after the decimal. So therefore, you can have a better control for your p-value threshold. So that means you're not thresholding it to be too stringent if you have a very coarse resolution of p-values. So in that case, the procedure would try to guarantee the FDR. If the p-value is not smaller or equal than the threshold, you just don't keep it. But it could be close. So in that case, you may miss a discovery. Okay, so this is just a very, very brief introduction, the first introduction to multiple testing. We will revisit this topic at I think, later part of our lectures. Okay, so we will move on to single cell. So this is a cartoon to give you a very bird view introduction. I think many of you have tried single cell data analysis from the 229 class last lecture. So it shouldn't be that new to you, but this time we will be more careful to talk about it step by step. Okay. So first of all, as it's sequencing, RNA-seq, right? Even though we move to single cell resolution, still RNA-seq. For RNA-seq, we will use next generation sequencing machine. For example, Illumina sequencing, which is the state-of-the-art sequencing machine. And what you get out of the sequencing machine is a FASTQ file. So Q stands for quality. It contains the sequenced short reads with sequences and the quality labels. So we would be able to judge which reads have poor quality and should not be kept in this step. So basically, the quality control step is here, the QC is for the read quality control. Maybe you will not keep everything in the FASTQ file, but now these things are typically done in the read processing software from the sequencing company. So you will convert the FASTQ file by mapping the reads to your reference genome. And mapping means that you do the sequence alignment. We compare each short read to the reference genome, especially for the species we study most, human and mouse. We do have very good quality reference genome, even though we're still improving it, but we can say it's good quality for most of the genes we're interested in. Then after the alignment or mapping, remapping, you would be able to count how many reads are mapped to each gene. And you would need some other steps, for example, depending on your technology, because for single-cell RNA sequencing, many technologies will use a technique called UMI to remove PCR amplification bias. So on a later slide today, I will talk about UMI. So just keep in mind that there is some differences in technologies. So the count here may be read count, or it may be UMI count. And you may also need to do some further filtering of cells in this step after you get a count matrix, which is what we will focus on today. So the quality control on the counts will try to remove the cells. So here the count matrix is gene by cell matrix, columns are counts. Try to remove the cells that are unlikely to be good quality live cells, but are rather like bad cells, which doesn't look like normal biologically functioning. So to remove those cells would help with the other steps, because starting from this step, the count matrix, if we believe it's good quality, containing good quality cells and informative genes, because you would also throw away the genes that are just consistently zeros in all cells, because that means the gene is not expressed at all and therefore not informative. So we would not keep it. And also we would define an arbitrary threshold to throw away the genes with too many zeros. Some people use, say, 95% zeros and more as the threshold. So you will not keep those genes. But for today, we will focus on filtering cells, the cells that are unlikely to be of good quality. So with the filtering, then we will go through cell clustering. That's a common analysis. And the purpose for this analysis is trying to give every cell a label. Because when we do the single-cell RNA-seq, we measure cells all together. We don't know which type each cell is. We just measure all cells in the sample, right? Like blood cells. It can have T cells, B cells, and red cells. So we measure everything. And then when we do clustering, we would try to find clusters as distinct groups of cells. So the clustering would totally based on this count matrix. So cells in one cluster are expected to share similar columns. Okay, and then once we're satisfied with the clusters, we still don't know the label of each cluster. So what do? We could run differential gene expression analysis. And the step here is trying to understand which genes are highly expressed in each cluster. And then we use our knowledge to match the highly expressed genes in this cluster to what we know as the marker genes of cell types. So for example, for CD4 T cells, we may know what genes are highly expressed. For B cells, we may know what genes are highly expressed. Then we just match the result from this step to our knowledge to give the cluster a label. So this is called cell type annotation. And of course, for the top route, this is from this genome biology paper, we may also try to do some gene regulation analysis. That is, given the genes, we can try to use some public database and algorithms to help us guess which transcription factors might be the regulators of this cell cluster. Like the transcription factors may be the regulator that activate the genes in this cluster. So that's called gene regulation analysis. So that's something we will not focus on, but I think this cartoon will give us a very good summary of the major steps from reads, quality control, mapping, to counts, After counts, we do a further quality control, then cell clustering, then cell cluster differential gene expression, then cell type annotation. Then we can say, okay, how many cell types are in the sample? What are their proportions? And we can look at the gene expression profiles for the non-marker genes. Our knowledge doesn't know, we can check their profile in single cell data. That's our new knowledge. Okay, so that's the whole pipeline. I hope it's clear. So for today, we will focus on the quality control step in Seurat. And this quality control is after we get the count matrix. So Seurat is probably the most widely used pipeline software package for single-cell RNA-seq data analysis because it's a multi-year group effort with many software engineers involved. So making Seurat, it's like an app store. It has almost everything inside, and because it's well-maintained, updated, now it's in version 5. It has become the standard. Like most people who are data generators, generate single cell data and do the analysis, they will use Seurat. So that's why I would use it for the teaching and also for introducing statistical concepts. Because given that people may use Seurat in your future research, I would say this is valuable. And I would say the counterpart of Seurat, because Seurat is our package, the counterpart for Seurat in Python is called ScanPy, but they were not developed by the same team. So therefore, their functions are not one-to-one corresponding because the options may be diverse. So therefore, for today, for this lecture, for simplicity, I'll focus on Seurat. But if you use ScanPy as a Python user, you will still find the topics relevant because the steps are similar, but the options in each step may be slightly different. Okay, so Seurat defines its own object class in R. So you would need to first create a Seurat package. But basically, the first step you need to load, and this is from the 10x genomics technology. And 10x is the popular commercial technology for single-cell RNA-seq. So many labs are using it, right? So here, UMI, unique molecular identifier, is what's generated. ScanPy. So yeah, thanks for answering. And so what we want to do here is to first reading a count matrix, okay, from this folder. So basically, you should be able to download this matrix PBMC data. PBMC is for the blood cells, basically. It's just a lot large. I mean, it's just many blood cell types contains, and it's the most widely used data type for many reasons. One reason is the ease of sample collection. It's easy to collect blood from a live person without much hurt. So for PBMC data, it's a count matrix. You load it into the Seurat function, create Seurat object to convert it to a Seurat object because Seurat has its own structure for it to use for other downstream steps. Okay, so we will first check the steps for single cell data analysis. This is from Seurat. Set up the Seurat object, which we already see from count matrix to Seurat object. Standard pre-processing workflow. This is basically the quality control, cell filtering we'll talk today. Normalizing the data, which we'll talk next, data normalization, identification of highly variable features, feature selection, data scaling, linear dimension reduction, which is typically PCA, determine the dimensionality of the data set, which is regarding how many PCs you keep. Then you cluster cells and you run nonlinear dimension reduction. UMAP t-SNE to visualize the cells in the 2D plot. And based on the clusters, but not the visualization, you will just use it for visualization, but not anything analysis, any downstream analysis on the visualization. So going back to the clusters, you want to find differential express features, the cluster markers. Finally, given the cluster markers, you assign a cell type identity to clusters. So these are all the steps we will cover in this quarter. So what is a count matrix? I think this is something good to know because in R, the count matrix, which is gene by cell, so the rows are genes, columns are cells. So this command will let us just show the first 30 cells. And sometimes you will see people say barcode instead of cells. The reason is in the single cell RNA-seq experiment, we barcode the RNA from each cell, hopefully from each cell, in a droplet, because 10x is droplet-based technology. In the droplet, a cell barcode is added, and then the barcode will be attached to the RNAs in the droplet. So then we will use the barcode to tell which RNAs are from one cell or from one droplet. But as we'll see later that a droplet may be an empty droplet, or it may contain more than one cell. And these are all the other issues we need to consider. But for now, you will see people may use barcode and cells exchangeably. So here we are just visualizing the first 30 barcodes or cells. All right. And regarding the data format, this is like a programming issue. You may store the matrix as a dense matrix. So dense matrix is just the regular matrix in R. So PBMC data, when it's loaded, it's in the format of sparse matrix, which is smaller than dense matrix because you just don't keep the zeros. For the zero entries, you don't record their values as zero. You just don't record values. You only record the non-zero values. When the zeros are the majority, then sparse matrix will be much smaller than a dense matrix. So this is just trying to compare when you convert this sparse matrix back to dense matrix, what's the size and what's the size of the sparse matrix. So it's like 24-fold more. So that's why it's good to know that in R, you have the sparse matrix format to use. Okay, for quality control, basically, this is what Seurat tells you. So Seurat allows you to easily explore QC, quality control metrics, and filter cells based on any user-defined criteria. A few QC metrics commonly used by the communities include, and this is from this paper, I think one of the early single-cell papers back in 2016. So I will share this as a paper for lecture 2 on Brunner. So you can check the original papers if you're interested. But basically trying to identify low quality cells by certain criteria. So let's just see here. So our approach is, so basically has defined 20. So in this paper, it actually defined 20 biological and technical features for you to filter out cells. And it shows several examples. So basically, you can see that in the old days, the cell number is about 5,000, but now it can get to, say, 100K in one experiment. Okay, so back to the Seurat introduction. The key features Seurat allows you to use for filtering out low-quality cells include the number of unique genes detected in each cell. So by unique genes, we mean that if you go back to this matrix, every cell is a column. You just count how many entries are non-zero in the column, and that's the number of detected genes. So maybe it's easier to see here, right? So here, if we just look at the 3 rows, cell 1 has 2 detected genes, cell 2 has 1 detected gene, and cell 3 has 2 detected genes. That's how we count the number of detected genes. Okay, and the idea for using this for filtering is that low quality cells or empty droplets will often have very few genes. And cell doublets or multiplets may have an aberrantly high gene count. So when you have 2 cells in one droplet in your technology, that's called doublet. When you have more than 2 cells in one droplet, that's called multiplet. But since the cells in one droplet will share the same barcode, then you would you would think they are one cell. But if you think, oh, they have an abnormally high number of gene count, this is called gene count, number of unique genes, this is called gene count, then you would think this is not one single cell. Then you may want to remove them. So in Surratt, it's very simple. As you can see, it will filter out the cells with a low gene count and high gene count. It will filter out the two ends. Okay, and another thing you can look at is that the number of molecules detected within a cell, which is correlated strongly with unique genes. So what it means is that you just take the sum for each column. So then here, the first cell would have a total count of 3, second cell total count of 2, third cell total count of 5, fourth cell total count of 10, and you just do the count. And we know that the more genes you detect, usually you have a higher total count. So that's why this feature and the first feature are likely correlated. Okay, and the third criteria is to count the number of reads mapped to mitochondria genes. So this is based on biology, the knowledge. We know that low quality or dying cells often exhibit extensive mitochondria contamination. It doesn't know. So that's why we will talk about this later. And the threshold, I can say that is arbitrary. So that's why many people, even though they use Surratt and Surratt has default thresholds, but on a particular data set, there's no guarantee that these filtering thresholds are optimal. So we will talk about the thresholding later. So we calculate the mitochondrial quality control. Surratt has a function. What the function does is that it will try to look for the gene names. As you can see that in the Surratt formula here, every gene has a name, right? CD3D, TCL something. So for the mitochondria genes in the reference genome, they will be annotated to start with MT. MT is the beginning of mitochondria genes. Then basically what Surratt does in this step is to look at the rows corresponding to mitochondria genes, and for each cell, add up the counts mapped to mitochondria genes. So then you will have a total mitochondrial count, and this percentage is just to calculate the percentage of the mitochondria gene counts among the total count. So it's like a mitochondria total count divided by the total count of all genes, and then convert that to percentage. So Surratt will do this step using its own function. Okay, then you can see that when you look at the Surratt metadata, so Surratt as an R object, it has a nice structure. It has this metadata to store what it outputs for the 3 features used for cell filtering. The total count, this is the total count called N count. This is the total, this is the number of detected genes called N feature, because Surratt regard every gene as a feature. And finally, it's the percentage of mitochondria gene counts. So you can see that the first 2 cells have quite high compared to the rest, higher mitochondria gene count. And the cell 1 has the total UMI count as 2,419, and number of genes as 779. So you can see that indeed the first 2 columns are quite well correlated. Okay, and Surratt can also provide this built-in violin plot. So you don't have to use ggplot2 for plotting it outside of Surratt. You can use the Surratt function just for this purpose. And then you are plotting the 3 features in the metadata, right? The metadata of PBMC. Then you just plot them as 3 violin plots to show the distribution. So the y-axis shows the value, and the curve, and by the way, if you're not familiar with violin plots, it's left and right symmetrical. So it's the same curve, just showing twice on both left and right. And the curve shows it's a horizontal, but basically it puts the horizontal density curve probability density function in a vertical way. So it's just showing, because the y-axis is the value. So what it says is that you have many cells whose total number of genes, whose gene count is around here, maybe around 700, 800. Many cells have gene count in this range, but you have few cells with very large gene count, right? These are outlier cells. And similarly, for total number of UMI counts, you see the similar trend. So for most of the cells, the total number of UMI count is close to, say, 2,000, but you have some cells with extremely large count. And people would guess, okay, these cells might be doublets or multiplets to be removed. And for mitochondria percentages, you can see that some cells have high percentages, higher than 5%, right? You'll see that. But back to the question, asked in the chat box by Daniel, you will see that there's no such clear-cut threshold. I don't know what you see from your eyes, but based on my eyes, I don't think there's a very clear-cut threshold. So you see a gap between the things you want to filter out and the things you want to keep. The change seems to be rather continuous. Okay. And you may look at pairs of features together, okay? This is just one features distribution. By feature, I mean number of genes, number, total count, percentage mitochondria, 3 features. But don't confuse this feature with this M feature thing. The M feature in Seurat refers to number of genes. So you can see that for the 3 features, we can look at their pairwise scatter plot. Again, Seurat provides this built-in scatter plot, so you don't have to go back to use ggplot to feature scatter. So you see that between N count RNA and percentage mitochondria, there seems to be a slight negative correlation. So for the cells with high mitochondria counts, right, these ones, they actually have very low total count for the UMI, which again indicate that these cells may be dying cells. They have few RNA but high mitochondria, so you can filter them out. Okay, and when you look at the number of genes and total count, you do confirm our intuition that they are strongly correlated. When one is high, the other is high. So therefore, using which one, N count or N feature, to filter out the cells with high gene expression that look like doublets or multiplets and the cells with too few gene expression, you may get similar results. So you can just use one of the two instead of both. And in Seurat, the default is this. You would only keep the cells whose number of genes is greater than 200 and less than 2,500, because you believe that above this, they are likely doublets, and below that, the cells may be dying. And you also set a criterion for filtering out cells with mitochondria percentages under 5. So you'll see then the line will be drawn here. 5 is here, right? These cells above it will be gone. And then 2,500 is here. So you will draw a line here and you will draw 200 here. So these parts will be gone and anything above it will be gone. So you will throw away those cells and they will no longer be in your count matrix as columns. Okay, that's in Seurat. But we also have some other references for you to 10x Genomics, which is the data, the kit company, the technology. People use the 10x Genomics kit for single-cell RNA-seq. And because it's more standardized than if you develop the experimental protocol in your lab. So we will go use the 10x Genomics website for further information. It has more criteria than Seurat. So it mentions that for the filters discussed in points 1, 2, 3 below, so we will go through them again just as a review. The filtering can be performed using various community developed packages, including Seurat and ScanPy. So ScanPy is here. Okay, for other filters and methods, we provide a few examples of community developed tools for researchers to explore. So first one, filtering cell barcodes by UMI count. So they're referring to cell barcodes because a barcode may not be a cell. The total UMI counts associated with the cell barcode represent the absolute number of observed transcripts in the droplet. Okay, so this is the definition from Wiki about UMI. They are short sequences or molecular tags added to DNA fragments in some next-generation sequencing library preparation protocols to identify the input DNA molecule. These tags are added before PCR amplification and can be used to reduce errors and quantitative bias introduced by the amplification. Okay, so we can say that when we use UMI counts instead of read counts, we remove PCR amplification bias. So here we continue from the description. Barcodes associated with unusually high UMI counts might be multiplets, that is, one droplet containing multiple cells, whereas barcodes with low UMI counts might be droplets containing ambient RNAs, just a floating RNA in the solution, in the liquid, but not real cells. Therefore, using UMI counts to filter cell barcodes may help to eliminate barcodes that do not represent a single cell. The choice of UMI count thresholds in published literature can vary between arbitrary cutoffs or the use of data-driven thresholds. That is, so in these two papers, you can check from the 10x website. The link is here. It's the URL. 3 to 5 times of standard deviation or median absolute deviation from the median. So what does it mean? So there are twofold information. So first, you calculate the median UMI count across all cells. And then you consider how high is high, how low is low. What you did is that you can compute the standard deviation of UMI counts across all cells. Then you consider median plus 3 times SD, standard deviation, or 5 times SD as the upper threshold. You want to throw away cells whose UMI counts are above that threshold. That's one way. Or you can go the other direction, median minus 3 times SD or 5 times SD to set that as the low threshold. If it's greater than 0, then you throw away the cells lower than that threshold. Another paper uses a different way, which is median plus median absolute deviation. So what does that mean? So mathematically, for every cell's UMI count, you calculate its absolute difference from the median. So that's called absolute deviation. Basically, you take the difference between a cell barcode's UMI count and the median UMI count. You take the difference and take the absolute value of the difference, so you don't care about the sign. And then for these absolute differences, you take the median. So then that is called the median absolute deviation. So then you will consider median plus median absolute deviation as the upper bound threshold, median minus median absolute deviation as the lower bound threshold. So that is just a way to help you decide which cells to throw away and to keep. But I would say it's still arbitrary, right? It's why 3, why 5, why not 2? So anyway, so I would say it remains a question whether we have a better principled approach to filter ourselves, but this is what's there. Okay, Cell Ranger is the software developed by 10x Genomics for pre-processing of reads. So you will use Cell Ranger to get a UMI count matrix, which you can input into Seurat. But Cell Ranger takes care of the read to count steps. So in Cell Ranger, UMI count is capped at 500 in the second step of cell calling. So basically, you will not get UMI counts lower than 500. This is already done in Cell Ranger. Barcodes with less than 500 UMI counts will not be regarded as cells. However, when the sample is highly heterogeneous, one threshold on UMI count for the whole sample may not always be suitable, as it may eliminate real single cells with very high or low RNA contents, for example, neutrophils. So what this suggests here is that you need to really understand your data. If you generate data, you need to understand the biological systems you study so that you don't use an unreasonable threshold for filtering out real cells. But if we go back to to Surratt, you can see the Surratt criteria for filtering doesn't even use UMI count because it's correlated with gene number. It only uses gene number and the percent for mitochondria. Okay, so continue with 10x genomics. This is the first step. This is like the N count. It corresponds to N count in Surratt. And this corresponds to N feature in Surratt. Filtering cells by number of features. Similar to the observation with UMI counts, barcodes associated with unusually high number of features might be multiplets, whereas barcodes with low number of features might be droplets containing ambient RNAs but not real cells. Some publications use arbitrary cutoffs, while some may use a data-driven threshold. That is 2 to 5 times of standard deviation or median absolute deviation from the median. The same idea as before, but here you work with number of genes or features instead of UMI count. However, when the sample is highly heterogeneous, one threshold on the number of features may not always be suitable because it may eliminate real single cells that express a high variety or low number of genes. Same reasoning. Okay, step 3, mitochondrial rates, which is also in Surratt. So an increased level of transcripts from mitochondrial DNA in cells has been associated with unhealthy cell states. It can also be a result of broken cells where the cytoplasmic RNAs leaked out of the cell while the mitochondrial DNA RNAs retained in mitochondria are captured in the single cell assay. For the thresholds on the percent of mitochondrial reads, some publications use arbitrary cutoffs, while some use a data-driven threshold. Again, 3 to 5 times of standard deviation or median absolute deviation from the median. It's worth noting that the expression level of mitochondrial genes can vary across samples. Same issue, sample-specific properties. For some cell types, like cardiomyocytes, expression of mitochondrial genes may have a biological meanings, and filtering cell barcodes based on this may lead to bias in the data analysis. So for each of the 3 steps in Surratt, it seems that the threshold using default might not work for some specific systems. So we should be careful and we should try to look at our data and try to understand the data. Okay, that's 4 steps for criteria 1 to 3 in Surratt. 4 to 6 are not something in Surratt, but there are community-based developed tools for this purpose. First, doublet detection. The presence of doublets or multiplets may confound the analysis as these multiplets can contain more than one cell from different cell types. In addition to using UMI counts and number of features per cell for filtering multiplets, there are a number of community-developed software tools for identifying multiplets in single-cell data. Many of the algorithms used in these tools generate artificial doublets by merging 2 droplets into 1 artificial doublet and calculate the doublet score by comparing the gene expression profiles of barcodes in the data with artificial doublets. Some examples of tools include Doublet Finder, Scrupulous, Solo. Setting the threshold on a doublet score for filtering can be subjective and data dependent. So it is recommended to check the doublet score distribution. As mentioned, the best practices of using Scrupulous. The benchmark study on computational doublet detection for single-cell RNA-seq. This is actually my former student Miles' paper with me, could be a good resource for understanding the performance. So I can quickly say that in our benchmark back in 2021, back then, we found Doublet Finder to work the best. So this is the paper for benchmarking we had. So the take-home message is that we use multiple criteria to evaluate doublet detection method based on downstream analysis, such as clustering and differential express gene identification. And the conclusion is that we think Doublet Finder is the best for detection accuracy, while this method, CXDS, is the fastest. So specifically, I'll just quickly go over the Doublet Finder methodology. And this is a table from our paper, what it actually does. First, it's in R, and it does PCA, my bad. So PCA for dimension reduction, it uses PCA because some other methods may not use PCA. And it has a guidance on threshold selection, which we think is the best part of it. So what it does, Doublet Finder, is that it generates artificial doublets by averaging 2 randomly selected droplets gene expression profiles, by averaging. And the doublet score of each droplet is defined as the proportion of artificial doublets among its k nearest neighboring droplets in the PC space. So for each droplet, we look at k nearest neighbors, KNN, and calculate the proportion of artificial doublets we generated. And the PC space is a result of PCA. The dimension must be specified by users. Okay, but the number of neighbors, K, is selected by maximizing the mean variance normalized bimodality coefficient. So what you want to have is that you just use the proportion as a doublet score. Every droplet has a doublet score. Then you can plot the doublet score distribution across all droplets. And you want to find for each K, the KNN in this step determines the proportion, right? So for each K, you get a set of doublet scores for all droplets. Then you plot the distribution of doublet scores. And when you change K, the doublet score distribution will change. And you want to maximize the bimodality coefficient. And this is the reference for this definition of bimodality coefficient. What it actually means intuitively is that you want the doublet score distribution to be bimodal, to have two peaks in its density. It's not like unimodal. And the bimodality coefficient just measures the strength of the bimodality. And the intuition behind it is that you want the distribution to be bimodal. So then you can say one mode corresponds to doublets and multiplets to be removed, and the other modality corresponds to the good cells you want to keep. That's the intuition. So basically, I think doublet finder is good because its K is selected, KNN's K is selected in this way, has a rationale behind it instead of an arbitrary K. There's a question, wouldn't some of the doublets be triplets, quadruplets, assuming doublets already exist in data? So if you think about the technology, how it actually works is that what 10x works is that in the experiment, it keeps shooting out droplets at a very fast speed. And then for the other side, you shoot out cells. So then you will try to make the cell meet the droplet, and then the cell enters the droplet. That's how the experiment works. So then the appearance of doublets is due to random chance. For example, the cells may not be perfectly dissociated, then the two cells attach to each other, or just by chance, two cells enter one droplet. But you can think that the chances having triplets or quadruplets will decrease, right? It will be smaller than having doublets. So doublets are fewer than singlets, then triplets are fewer than doublets. So that's why people mostly talk about droplets, because there are more than triplets. That's my understanding of this problem. The artificial doublet, yeah, that's a problem. So when you create artificial doublets, it's possible that it's created from two droplets that are not singlets. So what happens is that if the droplets are doublets themselves, then you merge them. What you create is not artificial doublets, but artificial quadruplets, right? But people think that's rare because you still believe the majority of your droplets are singlets. So that's why you think that the majority of the artificial doublets created in this way is reasonable. But something you could use is that when you create artificial doublets, you don't randomly select droplets, but you select the things that are likely singlets. That's something you could refine, I would say. Okay. Yeah. So, and later, so my former student Miles and I wrote this protocol to show exactly what we did for the benchmark. And we actually have an R package called doublet collection, in which we have a collection of these methods and some benchmark data set. So users can do the benchmark themselves, especially if they want to try a new doublet detection method, they could use the data set building to this package. One thing to note is that in this version, because it was published later and then the original cell systems paper for benchmarking. So in this version, we added this newer method called SC doublet finder, SC DBL finder. So we found that its performance is similar to doublet finder, but it's very fast. So that's why we think it's a good method to use, SC-DBL finder. Okay, so that's step 4, doublet detection. Step 5, empty droplet detection, the other end of the story, right? What if the droplets are like empty droplets? So in Surat, what it does is just to filter out the cells with too few genes, like N feature less than 200. And for doublets, Surat just filter out N feature greater than 2,500, right? So it's arbitrary threshold, but doublet detection and empty droplet detection are just more refined ways to remove these problematic droplets. So for example, here there's a method called empty drops, which actually derives an ambient profile based on gene expression profiles from droplets with small UMI count. Then cell barcodes in the data with a significantly different profile from the ambient profile are regarded as cells. Otherwise, it's considered as empty droplets. So it's a more complicated Bayesian method for detecting empty droplets. So you can check it out if you're interested. That's step 5. Step 6, remove ambient RNAs associated with barcodes. So in droplet-based method, it's possible that ambient RNAs are encoded in the droplet together with an intact cell. So in this case, it's called contamination. The droplet is not an empty droplet. It does contain a cell, but the UMI count is not accurate because it comes from both the ambient RNA and the RNA in the cell. So then our ambient RNAs could come from contaminants or RNAs released from unhealthy cells. As a result, the contamination from ambient RNAs can distort the UMI counting and downstream analysis of gene expressions. Several software tools have been developed to remove ambient RNA signal in the count matrix. So you're not just dropping cells, but you're changing the counts to change some counts that are unreasonably large to a smaller value, including SoupX, the CountX cell bender. And actually, Wei Jian, I know here in our audience, he developed something for this, right? So that was his undergraduate project, but I got to collaborate with his undergraduate advisor, and we work on something under this task, try to remove ambient RNAs. So hopefully your paper will be published soon, Wei Jian. Yeah. Yeah. So for the things to watch for, also from the 10x website, yeah, I think these are very important messages we should read and keep in mind. The impact of filtering and the resulting data quality can only be judged based on the performance of downstream analysis. Therefore, this can be an iterative process. It may be helpful to begin with permissive, remember the word permissive filtering approaches. So don't remove too many, don't be too aggressive, and then revisit the filtering parameters if the downstream analysis results cannot be interpreted. Okay, in some cases, it may also be beneficial to perform raw cell type annotation before filtering to avoid filtering out biologically meaningful cells. Yeah. There's not one set of thresholds for the metrics listed above that will apply to all data sets. Tutorials mention thresholds as starting places. For example, the Surat guided clustering tutorial, which we just looked at today, mentions arbitrary filters for the demonstrated data set. That is, filtering cells with unique features count over 2,500 or less than 200, and filtering cells with larger than 5% mitochondrial count. Publications often list thresholds that have been used based on the data. However, this does not imply that these are filtering methods that should commonly be applied to every dataset. The type and number of filters applicable to any dataset is highly dependent on the sample and cell type, as well as the biological questions. Different tissues or cell types may display distinct characteristics, such as heterogeneity in RNA content and mitochondrial gene expression. The same set of filters may not be appropriate for all cell types. In some cases, cluster-specific QC and filtering within the same dataset may be beneficial. That means for a certain cluster, if it represents a certain cell type, then the cells of that type may have some specific high gene expression or high gene count or low gene expression, low gene count. So that's what it means by cluster specific. If available, reviewing the literature for single-cell experiments with similar samples or cell types can help gauge filtering parameters that may be needed. Yeah, so it's something that needs careful analysis, not just going for the default. But I would say maybe for convenience, right? Many people may go with the Surratt suggested thresholds, and that's something I think people could do better. Okay, and finally, before we wrap up today, I want to share this paper called Current Best Practices in Single-Cell RNA-seq Analysis, a tutorial. It's not so new. It was published in 2019, but the steps in Surratt were mentioned in this paper. And this is from Fabian Thea's lab. I think his lab developed the ScanPy, and therefore they are in a very good position to do the summary. So again, the major steps are quality control, normalization, data correction, feature selection, dimension reduction. So people may say in different ways, but the steps are similar. So for quality control in this review paper, it has talked about similar points. So the quality control is based on these called QC covariates. In Surratt's called features, right? It's another jargon people use exchangeably. 3 QC covariates: the number of counts per barcode, count depth, the number of genes per barcode, and the fractional counts from mitochondria genes per barcode. Okay, so we have revisited this again and again. The distribution of these QC covariates are examined for outlier peaks that are filtered out by thresholding. So we'll see one example very soon. Next slide to see how the filtering is done in this paper. These outlier barcodes can correspond to dying cells, cells whose membranes are broken, or doublets. For example, barcodes with a low count depth, few detected genes, and the high fraction of mitochondria counts are indicative of cells whose cytoplasmic mRNAs has leaked out through a broken membrane, and thus only mRNAs located in the mitochondria is still conserved. In contrast, cells with unexpectedly high counts and a large number of detected genes may represent doublets. Thus, high count depth thresholds are commonly used to filter out potential doublets. 3 recent doublet detection tools offer, solutions, right? Including the doublet finder, which we think work well. Okay, so this is figure 2 from this paper, which shows how the thresholds were set. But I'll leave this for your judgment because to me, I have some questions. So this is based on the UMI count thresholding. So this is the distribution for all cells, for all cell barcodes. But looking at this, you may see, it's a unimodal distribution. Where do I set the threshold? For the low threshold, right? To filter out the empty droplets. Then they zoom in to the range from 0 to 4,000, this range, and show just the distribution in this range. And they pick this, which is close to 1,500. And the rationale, you see, a threshold at 1,500 based on the peak detected around 1,200. So it's talking about this peak. So pick this threshold here. But my question is, why don't you go here, right? There's a minor peak right here as well. So that's my question. Okay, and then for number of gene filtering, it's here. I think it's 700 genes. So for the droplets with fewer than 700 genes were filtered out. And here the rationale is a small noise peak is visible at approximately 400 genes. I think it was talking about this peak. But then my question is, why don't you go here? Go to the dip here instead of getting here. Okay, sorry. And then this is showing 2 QC features covariates plot together. Oh, no, sorry, my bad. Now it's showing the rank. So for the count depth, so in this paper, they use this count depth as the total count, while in the Surratt, it's the N count, okay? So for count depth, it just plot the cell the droplets from the one with the largest count depth to the lowest. So basically, you sort them. So that's why it's a monotone relationship. This is the top largest droplet, smallest droplet. And then it picks the count depth threshold which is 1,500, which is corresponding to here, by justifying that, okay, there's an elbow here. So it has a sharp decline here, elbow 1,500. And 1,500 is the vertical line in here. The last figure is to look at two QC features covariates together. Number of genes, count depth. So this is the N feature in Seurat. This is the N count in Seurat. Using the N count is 1,500. And this is the number of genes, which is the 700. So put them together. Put this threshold and this threshold together. You will only keep the droplets in the upper, this one, in the upper square. Basically, you only keep it there. But anything are filtered out. But if you look at them together, it doesn't seem to suggest like a sharp change point or anything, right? So it's pretty continuous. Anyway, so this is what's used in this paper. But in Seurat, it only based on number of genes. It doesn't use the count depth. Okay. So I think this gives you an idea about how arbitrary the thresholds are. Okay, and this is another message. Considering any of the three QC covariates in isolation can lead to misinterpretation of cellular signals. For example, cells with a comparatively high fraction of mitochondria counts may be involved in respiratory processes. Likewise, other QC covariates also have biological interpretations. Cells with low counts and/or genes may correspond to quiescent cell populations, and cells with high counts may be larger in size. Indeed, molecular counts can differ strongly between cells. See case study on project. Thus, QC covariates should be considered jointly when univariate thresholding decisions are made. And these thresholds should be set as permissive as possible to avoid filtering out viable cell populations unintentionally. In future, filtering models that account for multivariate QC dependencies may provide sensitive QC options, but it's not there. And quality control is performed to ensure that the data quality is sufficient for downstream analysis. As sufficient data quality cannot be determined a priori, it is judged based on downstream analysis performance, such as clustering annotation. Thus, it may be necessary to revisit with quality control decisions multiple times when analyzing the data. This is the iterative procedure mentioned on the 10x website. Often, it is beneficial to start with permissive QC thresholds and investigate effects of these thresholds before going back to perform more stringent QC. This approach is particularly relevant for data sets containing heterogeneous cell populations where cell types or states may be misinterpreted as low-quality outlier cells. In low-quality data sets, stringent QC thresholds may be necessary. So you need to have an idea about the quality of your data. And the quality of a data set may be determined by experimental QC metrics. And it has the table there. In this iterative QC optimization, one should be aware of data peaking in highlight. QC threshold should not be adapted to improve the outcome of a statistical test. So this is called data peaking, which is like you have an end result you're looking for, and you adjust your analysis to satisfy that. Instead, QC can be evaluated from the distribution of QC covariates in data set visualizations and clustering. So we'll end here. The last slide, pitfalls and recommendations. Perform QC by finding outlier peaks in the number of genes, the count depth, and the fraction of mitochondrial reads. Consider these covariates jointly instead of separately. Be as permissive of QC thresholding as possible, and revisit QC if downstream clustering cannot be interpreted. If the distribution of QC covariates differ between samples, QC thresholds should be determined separately for each sample to account for sample quality differences. Okay, so that's all for today, and I will post the slides and the related papers to the Bruner, so you can further check the result. But basically, we covered the first step for single-cell data analysis, and we will move to the next step in our Monday lecture. Okay, so that's all for today, and I will stop the recording.